We have to start with homeostasis. As we know, the life was started in marine environment, and the cells are structural and functional unit of life. So the cells were evolved in marine environment. So the in case of the multicellular organism, even these cells are same that were originated in marine environment. These cells have same uh, are similar requirements as that of the first or primitive cells evolved in marine environment. But in case of multicellular organisms, the cells are not in contact with the external environment, and most of them, they are also not in contact with uh, other cells, and they cannot function. These cells, they cannot function if they are not in contact with other cells. So the cells in multicellular organisms, they are in direct contact with the internal environment, and most of this internal environment is consist of water, and that water is called as the fluid, and this water is divided into two major portion, that is extracellular fluid and intracellular fluid. In human body, this body fluid is 60% of the total body weight, and 40% is dry matter. Out of this 60% body fluid, 40% is the cytoplasm or intracellular fluid, and 20% is making the extracellular fluid, that is outside of the cell. Intracellular fluid is that is inside of the cell, and that is in the form of cytoplasm. Extracellular fluid is in two forms. It is the plasma that is running inside the blood vessels, and then there is interstitial fluid, and interstitial fluid is more. It is 3 by 4 of the total extracellular fluid, and 1 by 4 is the plasma in human body. The plasma and extracellular fluid, that is interstitial fluid, they have more or less similar composition. We can say interstitial fluid is an ultrafiltrate of plasma, because plasma and interstitial fluid, they have the same small ions, but they have only difference of proteins. So protein, because larger proteins in blood, they cannot cross the capillary membranes and go into the interstitial fluid. They are more in blood vessel and less in interstitial fluid. So we can say interstitial fluid is an ultrafiltrate of plasma. Uh, these three portions of body fluid, they are uh, somewhat in contact, because we know the cell membrane, it is bounding the cytoplasm or intracellular fluid, and the cell membrane, it is differentially permeable, it is selectively permeable. So it will allow some substances to pass into interstitial fluid, or from pass from interstitial fluid into the cytoplasm, but not all the substances. So if we see cytoplasm or intracellular fluid, it is more concentrated with potassium ions, magnesium ions, protein, AMP, ATP, ADP, but they are very less in interstitial fluid, because membrane will not allow these substances to go into the interstitial fluid. However, in interstitial fluid and plasma, the ions concentration is somewhat similar, like there is equal concentrations of sodium ions, chloride ions, and bicarbonate ions, but there is only, as we said previously, only difference of larger protein. They cannot cross membranes, or we can say the one cell thick capillaries to go into the interstitial fluid. So the sodium, bicarbonate, and chloride, they are more in interstitial fluid as compared to the cytoplasm, because the cell membrane, it is not allowing these ions to freely go into the cytoplasm, so because it is selectively permeable. As we know, cytoplasm is important for the life of the cell, because it provides a medium for chemical reactions. It is for the transport of various chemicals, nutrients in the different parts of the cells. It allows mixing of these substances, help in every function of the cell. Similarly, interstitial fluid is important because it provides all the substances required by the cell, and it gets these material from blood vessels, from plasma, and then it send it to the, it is a medium between, it is a connection between plasma and cytoplasm. So it provides substances to cytoplasm or to the cell for the normal function of the cell. And it takes waste from, because the cells, they release waste into interstitial fluid. We can say interstitial fluid is working like a marine environment in which the cells were originated. So these cells, they release their waste into interstitial fluid, and they take their required substances from the interstitial fluid. So there, this interstitial fluid, it releases the waste released by cells into the plasma, and it gets the nutrients from the plasma for cells. Then plasma is important because it is carrying substances from outside of the environment into the body or the internal environment. We know we take food from outside of the environment, and this food goes into the digestive system, and it is digested, and the smaller molecule, they can get absorbed into blood, and then this blood is circulating in whole body and providing these substances to the cells, and taking waste material, that is carbon dioxide and the urea and other waste, to the kidneys. They take the plasma, it is taking this through veins, it is taking substances back from cell, that is the urea to kidneys, the carbon dioxide to the respiratory system to release them. The osmolarity of extracellular fluid is 290 milliosmol per liter, and the osmolarity is actually number of cations and anions per liter of the solution. So we can say this cation and anions, they put pressure to absorb water into that fluid, and this pressure is called as osmotic pressure. The force that is exerted by cations and anions in a solution to attract water is called as osmotic pressure. We can get this concept of osmolarity by using this diagram. We have a container that is having two compartments, compartment A and compartment B. Compartment A is fitted with a piston, and there is a semi-permeable membrane installed between compartment A and compartment B. Then if we put extracellular fluid in compartment A and simple water in compartment B, then we know the water will start flowing from compartment B into compartment A due to the osmosis. And if we have to stop entry of this water into compartment A, we need to push the piston on compartment A, and we have to exert a force. This force is equal to the force that is attracted, that is exerted by cation and anions on water. So this force is called as osmotic pressure or osmotic force. So if we have 1 liter of extracellular fluid, that is plasma or interstitial fluid, the cations and anions present in this extracellular fluid, they exert 290 milliosmol osmotic pressure on water to attract it into the extracellular. As we know, the interstitial fluid, the plasma and the intracellular fluid, they are in connection. The intracellular fluid, it is in contact with interstitial fluid through a semi-permeable membrane, and that is allowing some substances to go out or to come in and not allow others. The plasma is in contact with interstitial fluid through the thin 1 cell thick capillary membrane that have a gap in between the cells. These gaps, they are called as fenestrae or window, so the plasma can flow into interstitial fluid. These fluid, they are in contact. If the osmolality of one fluid will change, it will change the osmolality of others. If we consume more sodium, then it will go from digestive system into the blood, and it will change osmolality of the blood. So the water will come from interstitial fluid into the plasma, and the concentration of salts in the interstitial fluid due to the depletion of water will increase, and that will attract water from intracellular fluid into the interstitial fluid. Therefore, the extracellular fluid will increase and the intracellular fluid will shrink. If we have fever, then water will go out. It will be released from body, and the plasma will become thick. Its osmolality will increase up to 320 milliosmol per liter. So it will attract more water from interstitial fluid, and then interstitial fluid will attract water from intracellular fluid. So the intracellular fluid will shrink. That is the dehydration. And if conversely we drink more water, that water will come into plasma, and the plasma will become hypoosmolar. It means the osmolarity will go to 280 milliosmol per liter, for example. Then the water will enter into interstitial fluid and then into intracellular fluid to equilibrate. So the osmolarity will decrease. So the cells, they will swell up in case we drink more water, and shrinks they shrink if we drink less water or we have dehydration. If we drink isotonic solution that is having same concentration of fluid as that of extracellular fluid, then there will be no change in the volume of plasma, interstitial fluid and the intracellular fluid. But if we drink hypotonic solution that is having concentration of solutes less than the plasma, then the water will enter into interstitial fluid and then into intracellular fluid, so the cells will swell up. If we drink hypertonic solution that is having more concentration of solute as compared to extracellular fluid like juices or any other fluid that is having more salts, then the water will come out from interstitial fluid into plasma, and in order to dilute that fluid and in connection, the water from intracellular fluid will also come out, and the cells, they will get shrink. So many diseases, they do same thing. They either expand the water or body fluids, or they contract these body fluid. Contraction mean decrease in body fluid, and expansion mean increase in body fluids. If we have vomiting or diarrhea, then we lose isotonic solution, and the volume of the extracellular fluid decrease. If people, they have multiple urines, that is called as polyuria, again the same concentration of fluid is lost as that of extracellular fluid, so the volume of the extracellular fluid decreases. In this case, the blood pressure of the body is decreased, and the number of cells, that is the blood cells, increases as compared to the plasma. Similarly, in case of any hemorrhage, that is the breakage of any blood vessel, loss of fluid will occur in case of accident or any leakage. So the isotonic solution will lose, and the hematocrit will increase, that is packed cell volume. We will get concept of this in practical class. The packed cell volume is the volume of the RBCs as compared to the other cells and plasma in and it is shown in percentages. The normal percentage of RBCs volume is 40%, and it can increase or decrease due to decrease or increase in extracellular fluid, that is the plasma. If we consume salty substances like chips, then the salt concentration in plasma will increase. This will attract water from interstitial fluid and the intracellular fluid, and this will increase blood pressure, and it will decrease hematocrit due to increase in or the packed cell volume due to increase in the plasma water. So the cells can live and function only when they are bathed by extracellular fluid, and the concentration and the environment of that extracellular fluid should be compatible for the survival of the cells. So the cells, they must obtain nutrient and discharge waste to the extracellular fluid. If the cells, they are not in contact with interstitial fluid, that is the part of extracellular fluid, then they will die. They cannot live without this connection. So the term extracellular fluid was coined by Claude Bernard, and the term homeostasis was coined by Walter B. Cannon. So in order to study homeostasis, we need to define environment first. Environment is set of abiotic and biotic factors. The biotic factors, they include the other cells, the other animals or organisms, and abiotic factors are the presence of the gases, carbon dioxide, oxygen, and other gases, the pH, the salt concentration, the temperature, and many others. So the animals, they use different strategies to live in this environment. They may conform, they may regulate, they may avoid, or they may do allostasis. So the animals that they may use all these strategies for different variables, or they may use only one strategy for all the variables. So few of the animals, they are called as conformer. These animals, they could be unicellular or multicellular organisms. If they conform to their external environment, then they are called as conformer, mean they cannot regulate their internal environment. Their internal environment becomes as the external environment is. So the internal environment is not stable, it is not fixed, it will change with external environment. So the conformers, they, why they conform to the external environment? Because their structures are not specialized. They can work in a broader range of the factors of the environment. So, and there are few examples. For example, starfish, they can withstand salinity. Annelid worms, they can have little oxygen to live. So their cells, their structures, they can work in broader ranges of salinity and oxygen respectively. In case of regulators, their structures are specialized. They cannot function in a broader range of environmental factors. They need narrow ranges of temperature, pressure, nutrients, and these regulators, their internal environment is stable. It will not change with the change in external environment. It will remain fixed, and they regulate it by putting energy. They use energy to stabilize their internal environment. So the avoiders, they avoid the external changing environment. They regulate a little, but they avoid the external environment. So we are avoider for the temperature. For example, if the temperature increases, we avoid that temperature. We avoid sunshine. We go to the cool or cold places. And if temperature decreases, we put on more clothes, and we use heater. So we must be avoiding that environment. So in case of temperature, we are like avoiders. And then the allostasis or enantiostasis is the technique that is adopted by organisms. They change their one physiological variable to counteract a change in another variable. For example, if the pH, if it is increased, the oxygen will bind less with the hemoglobin, and if the pH is decreased, the oxygen will bind more with the hemoglobin. Sorry, that is vice versa, that if pH is decreased, less oxygen will bind with hemoglobin, and if pH is increased, more oxygen will bind with hemoglobin. So blue crabs, they decrease they increase their pH to increase the binding of oxygen with hemoglobin. That is called as enhancement, because they are increasing their pH in order to get more oxygen in brackish water. So we said the animals, they could be conformer for one variable, they could be regulator for another variable, they could be avoider for another variable, they could be allostatic for other variables. So they used different strategies for different variables of the environment. So homeostasis or regulation is the maintenance of relatively stable internal environment as compared to the external changing environment. So animal need their internal environment fixed or stable, because their structures or their cellular structures, they can work in a narrow range as we described previously. So they are called as regulator. So in multicellular organisms, the cells, they make up body systems. The body systems, they are used to maintain homeostasis for the cells for their survival. So this is a cyclic activity. If body systems cannot maintain homeostasis, cells cannot survive and they cannot maintain body systems. Environmental factors are seen that are affecting homeostasis. For example, these factors are amount of energy rich molecules like fuel, that is nutrients, oxygen and carbon dioxide concentrations for chemical reaction, waste products that are in the form of toxicity, pH, water, salt, electrolytes, volume and pressure of fluids and gases, temperature, they should be in narrow range, social parameter like other animals and cells, they affect the homeostasis of the regulators are the animals that maintain their internal environment. It means they maintain homeostasis. Their structures cannot work in broader ranges. Their structures can work only when the variables of the environment are on a single point, and that point is called as a. However, the variables like temperature, pressure, pH, solute concentration and others, they are not always on the set point. They fluctuate around the set point. The reason may be that the animal body is energy efficient, and keeping variable on set point needs more energy. So the animals, they let these variables to fluctuate to and fro, up and down of the set point, but in a normal range. The normal range of the variable is the range in which the variables, they fluctuate, but the structures of the cells or animals, they can perform maximum function. So the variables, they, there could be another reason that the variables that are fluctuating, because maybe animal body cannot maintain them all the time on the set point. So there are two reasons: either the animal, they don't want to maintain because they are energy efficient, they are they are um, conserving their energy, and the second, they their structures are not efficient because they have to maintain these variable on the set point. So they may be, they are inefficient, they cannot maintain the variable on the set point. So the variables, they are fluctuating in a normal range. So for example, the set point for human body temperature is 37 degrees centigrade, blood pressure is 120 by 80 millimeter mercury, the pH is 7.4. So it is not all the time at the set point. For example, the body temperature could be a little lower than 37 degrees centigrade, it could be a little high than 37 degrees centigrade, but they would not go beyond 37.5 or 36.5. They will fluctuate in that normal range. A basic feedback system is a system in which the products can control the production of products. There are two types of feedback system: positive feedback system and negative feedback system. In positive feedback system, the products of our system can increase the production of products. In negative feedback system, the concentration of products can decrease the production of products. Same is with the chemical reactions that are positive feedback reaction and negative feedback reaction. In we know there is a rate of reaction. Rate of reaction is defined as the conversion of reactants into products per unit time. So in case of negative feedback reaction, the rate of reaction is decreased by the products. In case of positive feedback reaction, the concentration of products increase the rate of reaction. In homeostasis, most of the time it is the negative feedback system that works, and it maintains the variable. It brings variables when they are going away from the set point back to the set point and keep them in a normal range. A basic feedback system has three components: a sensor or a receptor which monitors the value of some variable, and then there is another thing that is called as integrator, or it is called as a control center, that assess the change in the variable, and it has the set point fixed in it. And then there is another component that is called as effector. So the effectors, they change the value of the variables under the instruction of integrator. They keep the variable near set point. What the effector do when they receive information from the control center? They introduce an antagonistic mechanism that opposes change in the variable. For example, if temperature falls, effector produce change to increase temperature. For example, in case of animals, the muscles that produce shivering to increase the production of heat, or the effector, they can change behavior of the animals. The animals, they seeks out a different location, they avoid that location, and for example, in the butterflies, monarch butterflies, they migrate to avoid the cold north. Here we can see stepwise maintenance of homeostasis in animal body. For example, any variable, temperature, pressure, pH, is fluctuating along in a normal range along set point, and if it goes up to above the normal levels of the normal range, then this change is called as a stimulus. Stimulus is any change in environment, normal environment. So this change of variable above the normal range is detected by receptors, and then these receptors, they send this information to the control center. In case of animals, it is either brain or spinal cord, and then these control center, they send signal to effector. Effectors in case of animals are muscles, and then these muscles, they contract, and the effector could be glands also. So these glands are muscles, they perform function, they anticipate that change, they negate that change and bring the variable back toward the set point in order to keep it in normal range, so the functions could be performed optimally. Similarly, if variable they are fluctuating in normal range and then by chance they are going down than the normal range, then this change again is a stimulus that is detected by receptors present over there. These receptors they again send information to the control center. Control center has the set point for this variable, so control center will compare this change with the set point and then the control center will order to the effector, that are muscle or glands, to perform their function to negate that change again, to decrease that change again and bring the variable back to the set point. So it is called as negative feedback mechanism that is maintaining homeostasis. Now we have an example of maintenance of homeostasis of blood pressure. We know the blood pressure has set point at 120 by 80 millimeter mercury for a human being, and the blood pressure is dependent on three main factors. It is dependent on heart rate or force of contraction. We know the heart rate is frequency of heart per unit time. If frequency of heartbeat increases, then the blood pressure increases, and if frequency of heartbeat decreases, then the blood pressure decreases. Same is for the force of contraction. The diameter of blood vessels, if it increases, the blood pressure decreases, and if diameter of the blood pressure decreases, then the blood pressure increases. And the blood volume is uh, again directly proportional with blood pressure. If blood volume is increased, the blood pressure increases. If blood volume decreases, the blood pressure decreases. So these factors they are again controlled by various other things, or we can say factors. The heart rate is controlled by cardiac muscles. These cardiac muscles they are having their control in medulla oblongata, the part of brain, because they are involuntary, so they are controlled by medulla oblongata. Medulla oblongata can increase heart rate and force of contraction, or decrease heart rate or force of contraction. The diameter of blood vessels is dependent on smooth muscle or contraction of smooth muscles. So these smooth muscles they are again involuntary, their control is in medulla oblongata. So medulla oblongata is increasing diameter of blood vessel or decreasing diameter of blood vessel by contraction of smooth muscles or relaxation of smooth muscles. The blood volume mainly is dependent on two hormones, that is antidiuretic hormone and aldosterone. Antidiuretic hormone is released from posterior pituitary, and this antidiuretic hormone retains water in blood. It is antidiuretic, mean it is against production of urine. So this antidiuretic hormone it acts on the nephrons and it decreases the release of water in urine and increases blood volume, and in turn increases the blood pressure. Same is for aldosterone. Aldosterone is a steroid hormone that is released from adrenal glands, and these aldosterone it is also called as mineralocorticoid because it retains sodium in blood at nephrons, and when the aldosterone retains sodium, the water get attracted and it comes in plasma and it is not released in urine, so the blood volume is increased and blood pressure is increased. So in order to maintain blood pressure, the control center or medulla oblongata has to control all three factors, that is heart rate, diameter of blood vessels and blood volume. Here we can see the blood pressure is fluctuating in normal range along set point, that is 120 by 80 millimeter mercury. If by chance it goes beyond the normal range, above the normal range, then this change is detected by the receptors in blood vessels that are near to heart. These blood vessels are carotid arteries, and they send signal to or information to control center medulla oblongata, and then medulla oblongata send information to effectors. In this case, the effector could be muscles and the glands. The muscles are cardiac muscles in order to decrease the heart rate and the force of contraction in order to maintain blood pressure. And then to the smooth muscle control center also controls smooth muscles of the blood vessel to decrease the diameter of the blood vessel by relaxation of those muscles. So the blood vessel diameter of the blood vessel increases and the blood pressure decreases. And then the medulla oblongata, it also sends signals to pituitary and the adrenal gland to not release antidiuretic hormone and aldosterone, so the water is not retained in body, it is released in the urine, and blood volume should decrease in order to decrease the blood pressure. So this mechanism has decreased the change, it has negated the change. The change was increase in blood pressure, now this change is negated and the blood pressure has come back to the toward the set point, that is 120 by 80 millimeter mercury. Same is the case when the blood pressure decreases beyond 120 by 80 millimeter mercury. That change is detected by again baroreceptor in carotid arteries, and then these baroreceptor they send signal to the control center, that is medulla oblongata, and then medulla oblongata sends signal to the effectors, that is the uh, cardiac muscle, to increase the force of contraction and the frequency of contraction. The medulla oblongata also sends signal to the smooth muscles in uh, blood vessels to contract, to decrease the diameter of the blood vessel so that the blood pressure increases. And they also send signal to glands to release antidiuretic hormone and aldosterone so that more water is retained and the blood pressure should be blood volume should be increased and the blood pressure should increase. And so this system has negated the change. The change was decrease in blood pressure. Now this decrease is anticipated and it has the blood pressure is going towards set point. So negative feedback is maintaining blood pressure by negating the change, that is the blood pressure either going up than the normal range or going down than the normal range with the help of receptor baroreceptors that are in carotid arteries, control center that is in medulla oblongata, and effector that are smooth muscle, cardiac muscles, and pituitary and the adrenal gland. As we know, the maintenance of homeostasis is responsible for the survival of regulators that are the animals that regulate their internal environment, and the system that is maintaining homeostasis is negative feedback system. But negative feedback system has flaws that risk the life of animals. The first one is that the negative feedback system cannot predict a change in variable. The variable should actually be changed before the negative feedback system starts to work, so that delayed the response. And another thing that negative feedback system has is to overshoot the set point. Mean when the negative feedback system is bringing variable back to the set point, it will not stop at the set point, it will take variable to the other extreme of the set point. That is called overshooting the set point. For example, the blood pressure, if it was decreasing, the negative feedback mechanism will bring back to the set point, and then it will not stop at set point, it will take it toward the increased limit of the set point. So these flaws, they should be overcome. These we have a simple antagonistic or the negative feedback mechanism system. This system is made up of sensors that are detecting the variables all the time and sending information to integrator or control center. This control center has set point in it, and it is always comparing information of variable coming from sensor to the set point. If there is a difference, then the integrator or control center instruct the effectors to increase the variable or to decrease the variable, as the case may be. And we see this negative feedback mechanism or antagonistic mechanism cannot predict a change in variable. It will work only if there is an actual change in variable, but this can risk the survival of animals. So the nature has another system to help negative feedback system to predict the change in variable. This system is called as feed forward system or the anticipatory system. The feed forward system and anticipatory system gives two benefits: that it predicts the change before it occurs and in the variable, and it also does not allow the overshoot of the set point. So in case of feed forward system, the negative feedback mechanism or system that is made up of sensor, integrator and effector is helped by the anticipator that is getting information for other variable through its own sensors. So we can understand this by an example. For example, if we take a meal, that meal is converted into sugar, and the levels of blood sugar increases. That is detected by the receptors in blood vessels, and they send information to the integrator to release insulin from pancreas to adjust this sugar into muscle cells and the liver cells. But if the negative feedback mechanism would be working alone, then the increase in sugar would occur first, and then insulin would release. That may would have harm the body and would have wasted the sugar in urine. In case of actually this negative feedback mechanism in animal body is helped by feed forward system. That is, when we see meal, our sensors, like or we take meal in our buccal cavity or we ingest meal, the information is sent through sensors like eyes, from touch receptor, from nose to the anticipator, that sends signal to the control center to release the insulin before the increase in sugar actually occurs. So it is anticipating, it is predicting, the feed forward system is predicting that change is going to happen. So it is helping the negative feedback system to release insulin before the time. It is also helping to not allow the overshooting of the set point, because for example, in this case, if insulin is released more and the sugar is being decreased, it can go decrease than the normal values, but the feed forward system again detects it, and it helps the integrator to stop the release of insulin, so that the normal values of the sugar must be retained in blood. There are various other control systems in animal body. They are called as a rheostasis, that is non-homeostatic regulation. These regulations, they change the set point temporarily, permanently or cyclically. For example, in fever, the temperature of the body is increased. That is due to increase in set point. It is not the normal fluctuation, or it is not increase than the normal value. It is the actual shifting of set point above than the 37 degrees centigrade, that is the normal set point. The set point for the temperature is in hypothalamus, and there are receptors in blood vessel. These receptors, they are called as thermoreceptors. So thermoreceptors are detecting blood temperature, and they are sending signal to hypothalamus, and hypothalamus is comparing the actual temperature of the body to the set point. If the temperature is more than the set point, then that is in normal condition 37 degrees centigrade, then hypothalamus sends signal to the blood vessel on the surface of the body to relax, to increase their diameter, so that more blood flows on the surface and heat emits out. And if the temperature is decreased than the set point, then the hypothalamus sends signal to muscle to shiver, to produce more heat, to the blood vessels on the surface to decrease their diameter, so that less blood flows on the surface and the heat is retained in body. But a fever, the set point, that is 37 degrees centigrade, is increased up to, for example, 38 degrees centigrade, so that the body thinks that the temperature is down in body, and it is increasing it, and it brings it to the new set point, that is 38 degrees centigrade. So the temperature of the body is increased. Same is for the sex hormone concentration at puberty. In case of the prepubertal ages, the sex hormone concentration is less, and the set point is set to less levels. But at puberty or after puberty, the set point of the sex hormone increased, so that a new normal range is formed along that set point. Same is for the reproductive cycle or menstrual cycle. In this case, the female estrogen and progesterone, their set point is changed cyclically. During menses, the set point of the estrogen is decreased, and during cycle, the set point is increased. There is another control system that is called as positive feedback system. So positive feedback system, most of the time, it increases the change, and it takes variables away from the normal range or the set point of the variables, because we know the positive feedback reactions are those reactions that increase their product, increases the rate of reaction. For example, the blood pressure is increasing, then the positive feedback mechanism will increase that blood pressure. So this may harm the survival of the individual. But in few cases, the positive feedback mechanism is helpful. For example, in case of action potential, in case of lactation, in case of blood clotting, and in case of parturition, the positive feedback mechanism is helpful. But in most of the cases, the positive feedback system takes variable away from the set point, and it causes the death of the organism. And it, for example, in case of bloodshed or the during accidents, most of the deaths, they occur due to positive feedback system, because we know the heart muscles, they need nutrients and oxygen for their function, and these nutrients and oxygen, they are provided to heart muscle through coronary arteries. And if there is a blood loss due to accident, the volume of blood is decreased in blood vessel, that will decrease blood pressure. So the blood pressure also decreases in coronary arteries, so that the heart muscle, they are not getting optimal nutrients and oxygen, so they cannot work optimally, they cannot contract with normal force of contraction and frequency of contraction, so blood pressure decreases again. And if blood pressure decreases again, the the blood pressure decreases in coronary artery, and the heart muscles are not getting optimal food again, and they contract with less frequency and force, so that the blood pressure decreases more. So there is a cascade of reaction that will lead to the death of organism. So here we can see positive feedback mechanism has same system as that of the negative feedback system. It has sensor to detect the deviation in control variable, and they have the integrator or regulator or control center to compare the change with the set point, and this control center, it send instruction to effector to increase the change, to enhance the change, so that this system works in a vicious cycle, like we saw in previous example of blood pressure due to loss of blood volume, and then there would be ultimately there would be a death. But in few cases, like in birth, there the positive feedback mechanism is used. Birth is an example of positive feedback mechanism. Near the end of pregnancy, the baby's larger size stretches the muscles of uterus. This stretch is detected by receptors in uterus. Those receptors are mechanoreceptor. They send signal to hypothalamus, and hypothalamus sends signal to pituitary to release oxytocin. Oxytocin is an endocrine hormone, and it goes into the blood and reach to the muscles of uterus and help them to contract. This contraction pushes the baby in the next segment of the uterus, so that next segment of the uterus, again the muscle over there, they get stretched, and they send signal to the hypothalamus and again to pituitary, and pituitary releases oxytocin. Oxytocin again goes to the blood and then to the uterus muscle of that segment, and those muscles, they get contract due to oxytocin, and fetus is pushed in the next segment of the uterus, so that there would be birth. Sometimes whole range of the variable is shifted above or below the normal range. For example, in case of exercise, the blood pressure, the normal range of blood pressure is shifted above, and then at rest again it comes back to the normal level.